About 25 years ago, a large number of scientists from around the world got together and they had a pretty audacious and aspirational goal. They said there's about 3 billion base pairs, little chemical trains connected to each other that make up the genome. And they said, let's sequence it. A very bold initiative. $3 billion later, and about 12 or 13 years later, the human genome was sequenced. And that meant that the map of the human genome was for the first time accessible to scientists and therefore hopefully to doctors who could use that information to improve the prevention of disease, the diagnosis of disease and the treatment of disease. And quite simply, it has had a transformative effect on medicine. It's still got a long way to go, and I know we're going to hear from Professor Mitchell about that, but the reality is what it's done for science has been transformational in real terms. You can go and have your sequence performed of your genome for about $2,000, and we'll hear more about is that a good thing or not a little bit later on. But just very briefly, what is the Perkins doing about the genome? Inside the Perkins, as Larry Ifler has just said, we've got hundreds of people working on various aspects of human disease. Mainly the things that are going to kill us. Heart disease, and don't forget heart disease I'm saying first because it is still the biggest killer. Cancer and diabetes, and of course some other diseases such as rarer muscle and nerve diseases. And an example is Professor Nigel Lang. I'm not sure if I can see Nigel here. Don't think he's here. He's up there, is he somewhere? Oh, there he is. Oh, he's with his computer. Well, he's working. So Nigel Lang is the head of the neurogenetics unit, and he's identified about 25 different genes related to human disease in the last few years. A wonderful exemplar of using the genome and transferring that into human patients. Those patients with unusual diseases have now, for the very first time, got a diagnosis that helps them move towards potential therapies. Professor Alistair Forrest in the Perkins is setting up a wonderful and very sophisticated set of machines that will allow us to do what I call the master chef of DNA. It will allow us to deconstruct cancers into their single cells, which, believe it or not, might sound an audacious goal, but it is an important goal to deconstruct tumours into single cells, understand how the single cells work together, and then reconfigure that with new therapeutics. So the genome is having a transformative effect around the world, a transformative effect right here in Perth, and allowing us to do amazing research. So with that segue, I'm going to introduce Professor Christina Mitchell, who I've known for quite some time, and we've caught up this afternoon and had a good old chinwag and heard about some of the wonderful things that are happening in Monash. And she won't like me going into detail, but I'll spend a couple of minutes doing that. So a very bright graduate from the University of Melbourne, did medicine, and then always loved teaching, but always loved research. After becoming a doctor, became a haematologist, which is a blood-based specialist, treating leukemias and other disorders of the blood, and then loved research and did a PhD in Melbourne with Haddam Salem, and then, as most of us do who like science, went across to the University of Washington, went across to the United States, to the University of Washington, and did what's called a postdoctoral fellowship over there. Came back to Melbourne, and that's where she's really, as it were, gone into overdrive, because Christina has literally transformed the Department of Biochemistry at Monash Medical Centre and Monash University into this massive 2,000-person department that turns over many, many millions of dollars a year, and is probably the biggest and most prestigious department of any type in Australia, in Australian science. Christina became the first GO8 woman dean or female dean of medicine some years ago and is a leading light in academic medicine. She has run a laboratory working on signalling between cancer cells and inside cancer cells for many years and is an absolute leading light in that. So not only a leadership role from driving and the growth of a massive department in Monash University, but also driving her own research. She won the Lemberg Medal, which is the highest honour for the Society of Biochemistry of Australia. She is an honour roll member of the Victorian government, honoured for her service to medical research as a woman, and quite simply as an outstanding individual with a great sense of humour and always fun to be with. So, Christina, we're absolutely overjoyed you're here, and we look forward to hearing you talk about precision medicine. Please welcome Christina.
Introduction. I'd like to thank Peter and the organisers of this really prestigious lecture, and the Charles and the Perkins Institute for this wonderful opportunity. So the Perkins Institute. Maybe it'd be better if I had it turned off. The Perkins Institute is a wonderful institute. Peter's done a wonderful job. I'm inspired by what you're doing in the institute. I think you're making a real difference. It's creating a culture of innovation and translation, which I think is going to be for the great benefit of all Australians, not just for Western Australia. And it's a great honour to be with you here tonight. So I'm going to talk about precision medicine, which is the great hope and excitement for the future. And it's a term that's really become common in the lexicon over the last 10 years, in part stimulated by President Obama in his address. And precision medicine is an evolving strategy for disease prevention, diagnosis and treatment that incorporates the individual's genetic background, environmental influences with their clinical outcomes, which is tailored to improve treatment. So the aim of precision medicine is to lead to more accurate diagnoses, more rational disease prevention, and better treatment selection for the individual, not just for a whole population. So it's targeting medicine to an individual person based on their own DNA. So it's a massive challenge, and it's the big excitement in medical research today. So its current definition is understanding disease at a deep level, based on your own genetic material, which will lead to new targeted therapies. And it requires a whole new set of diagnostic techniques that are currently being evolved, including being able to sequence all your DNA or all your tumour DNA, and then with accuracy and precision say this is the therapy for you based on your DNA. That leads to a whole issue about this massive explosion of data and how we actually use it, and has led to a whole new industry of precision medicine, data sharing, smartphones to capture your data, and all of you going to Wikipedia to work out, and Google to work out, can you treat and diagnose yourselves, and do we really need doctors anymore? So it was actually President Obama who unveiled the Precision Medicine Initiative in the United States in his annual State of the Union address to the nation in January 2015. And before then, a lot of people, including a lot of doctors, had never heard of precision medicine. As yet, President Trump hasn't revoked this. And it's a huge initiative which is to study 1 million Americans and to sequence the DNA of 1 million Americans. And I'll explain to you in a minute what the scale of that is in terms of the data, to sequence the whole genome of 1 million Americans and then follow them and see what diseases they get, and then to go back to all that genetic data and say, well, they got this disease because they had this genetic variant. Everyone who participates will be a volunteer. They will donate their RNA, DNA, it will be sequenced, and then it will be linked to what's called an electronic medical record, which they have much more in the States than we have here. An electronic medical record is just being introduced in Victoria. I'm not sure if you have it in Western Australia yet. So that you can link that genetic data of the whole sequence of the individual with their clinical history and then draw all the patterns up together. So it's a longitudinal, long-term study. So this is a very massive, ambitious task. So this is taken from the National Institutes of Health, which is on the website, which anyone can go and look at. And basically, Obama defined precision medicine as an emerging approach for disease prevention. So if we can identify by looking at these 1 million Americans what their DNA looks like and then track to see which diseases they get, then perhaps we can say this variation in the DNA will predict for this particular disease. It also, then if we can match the genetic variations with the disease, then potentially we can design new treatments and get what's so-called targeted therapies. And with the long-term aim of bringing this into clinical practice. And it already is in clinical practice to some extent, and I'll give you some clinical examples. The other thing that they talk about, which is very interesting in the American context, is that they want to test whether mobile devices can encourage healthy behaviours. So can having a mobile device which, you walk your 10,000 steps, or you climb your stairs, and then you link it with your genetic data, and then you look at your long-term health outcomes, and can you capture that all on your mobile phone, and then get a little signal to say, walk some more steps or do whatever, is the long-term outcome. Now, this is a very ambitious target to use mobile devices, and we're not doing that in Australia yet. The other thing I'm going to talk about very briefly is the study of pharmacogenomics, which is looking at your genetic background and seeing whether you will have a different reaction to certain medications. So I'll mention that briefly. So it is a massive project that's being undertaken in the US. We have some small-scale projects similar to that, which I'll talk to you about, that we're undertaking at Monash University, which is looking at an entire population, sequencing all of their DNA, and then looking at long-term outcomes. The scale of the data I'll explain in a minute. At the same time, in the United Kingdom, in 2015, they announced the 100,000 Genomes Project, which is a project which is actually, unlike the project in the United States, which is completely funded by the government at the National Institutes of Health. The 1,000 Genomes Project is a very interesting project in which industry is actually partnering with great universities like Oxford and Cambridge to invest up front in sequencing 1,000 UK individuals in particular areas. So unlike the US 1 million genome project where they're sequencing a million normal Americans, they're going to sequence the genetic information of 100,000 British patients to study rare genetic diseases, including congenital hearing loss, as well as various cancers like breast and colon cancer. In addition, the US are actually doing a targeted sequencing strategy for cancers as well. So there is this massive large-scale sequencing projects going on to try and map changes in DNA with disease. But there's a problem. It's a problem that will be solved, but the data involved is absolutely enormous. So everyone's heard of DNA. So the DNA is your blueprint for what you will be like. You inherit it from your parents. There are 20,000 human genes. And as Peter said, which are encoded by 3 billion base pairs. So every time you sequence your whole DNA, you're looking at 3 billion base pairs that you have to sequence. In addition, 99% of it does not encode for protein. We don't know what a lot of the DNA does. But although you probably conceptually think that, you know, we all have DNA, therefore it's all the same, it's not. There are areas within the DNA which are called variants. So 1 in every 1,000 of those 3 billion base pairs is different between you and the person sitting next to you. There are 3 million variants in any one individual's DNA from another. So when you're sequencing 1 million people in the US to get 3 billion base pairs, and you're going to compare all the variation between all those million people and then work out what's causing a disease, the scale of the data is enormous. But they're doing it, and they will do it, and we will get the answers, but it requires massive computing and massive data sharing and analysis that is going to take years to work out. But with the rapid advances in technology, it will lead to rapid discoveries and new treatments. So if we look at DNA that is in all our cells, that's called germline DNA. If you have an inherited disease like cystic fibrosis or Duchenne muscular dystrophy, you'll be born with changes in your DNA that will lead to that disease. If you are completely normal and healthy, DNA will be fine, although there'll be variance between you and other people. But over a period of time, as you age, and if you smoke, drink, go out in the sun, live, get older, environmental, your DNA may acquire changes, DNA damage, mutations. And there's no doubt that as we get older, the incidence of cancer increases. So you may get mutations in the DNA, which will occur in some cells, but not all cells. So if you get cancer of the lung, you've probably developed a mutation in some of your lung cells' DNA, but won't be present in any other cell. And this may lead to changes in the cell that make it grow faster and lose its normal constraints on cell growth. So you get increasing DNA damage as you get older, and certain things such as UV light. We've got the highest incidence of skin cancer in the world. I imagine in Western Australia you're as high as anywhere else in Australia. And direct sunlight damages DNA in the skin, which can cause skin cancer. So when we have a genome project where we sequence normal people, we're sequencing this germline DNA, and when we get mutations in the DNA in cancer, we sequence the cancer DNA to compare the differences between the two, because the cancer DNA will have more damage in it than the germline DNA. So when we diagnose a cancer, we are now sequencing the cancer DNA to look for the differences between the cancer DNA and the normal DNA. So this is what it looks like when we do a sequence. We have this repeating base pairs, and we can see a difference when we have a mutation. A G here goes to a C, and when the DNA repair and copy makes an error while copying the DNA, it can make an error or a mutation. If that happens in one of the 19,000 genes that's not important and it doesn't make any difference, but if it hits a really important gene that's going to make the cell grow faster, then it can, if you get an accumulation of DNA damage, lead to increased cell proliferation and potentially cancer. So there's all sorts of sequencing mechanisms being described. You can sequence the whole genome and get billions of base pairs, or you can only sequence bits of it. So what we do in cancer is we tend not to sequence the whole genome, but people are offering services now where they can sequence your whole genome. I think you really need to ask, what are you going to get out of it? And do you want to know? And can anyone actually interpret it? So those are the questions that you should be asking. So you can sequence the whole genome, or you can sequence bits of the genome, but the end is you get an enormous amount of data, there's a slow turnaround time, and it requires extensive analysis. So it's who's got the expertise to analyse that. So we have DNA here, which can be, we test for this by doing DNA sequencing, looking for mutations. We tend to do this particularly in tumour DNA and with some inherited diseases to try and work out what the cause are in germline. That can be then transcribed into mRNA, and we can sequence that as well. So the DNA is the blueprint or the architectural plans that tells you what you're going to be like, and the proteins are what is the machinery or the motors of the cell, and we can actually work out what the proteins are doing by doing a technique called proteomics. So there's all these ways to measure what's going on. There's sequencing, RNA-seq, proteomics. There's a huge industry in this. So this is some of the things people do. They do all genomics, transcriptomics, proteomics, and it's called the omics industry. And then you end up with biomarker and disease detection, and the long-term aim is to look at new drug therapies, and this is precision medicine, and it's a massive industry. So I love this cartoon. Down the bottom it says the human being, some assembly required. And here is someone reading acres and acres and acres of DNA trying to interpret it. And I have this feeling he's just seen a mutation there and he's trying to put it all together. So why is this all now possible where it wasn't possible before? So as Peter said, the Human Genome Project's made a huge difference. But in actual fact, we're really yet to capitalise fully on the Human Genome Project, but it is coming because it took years to do the first human genome. Now you can sequence the genome in 2 weeks. But the issue is how do you interpret all the data? You can sequence it, and in fact, people are buying millions and millions of sequencing machines and churning out the sequencing. But really it is the cloud computing and the storage of the data. So with cloud computing, you can now share the data and analyse the data, which was not possible before. This is leading to a concept called network medicine. So you get all the data from the sequencing, and then people from all over the world can look at the data and analyse it and interpret it. One of the important things, because Peter and I are clinicians, we don't believe, at least I don't believe, that DNA by itself is enough. And Peter and I were having a very deep conversation about the fact that DNA by itself is meaningless unless you have the clinical history and outcome in the patient. It's uninterpretable. So we believe that you can get the DNA sequence, but you've really got to have a patient there and follow the outcome of the patient and correlate the two together to have a meaningful outcome. But if you can link electronically the DNA sequence with the history of patients from electronic medical record and use medical informatics, which is linking the two up using cloud computing, then you've got a very powerful tool. Now, one of the really important things that Obama mentioned in his precision medicine is that the patient and the individual has to be a willing participant in this, because your clinical record is your clinical record. You've got to be willing to share it. So the whole concept of precision medicine when you're doing whole population is that the volunteers are willing to share their data and share their clinical history and their DNA data to volunteer so that we can work it out by network computing. So Obama emphasised that in his precision medicine, and I think it's an issue that we need to think about, that this is all this data is networked by cloud computing. And the ability to capture and analyse massive data sets and to share the data. So with cloud computing, someone from the US and Australia can be working on the same data and sharing the data. It will be de-identified, but you can all work on the data and share it. So I believe precision medicine starts with the individual. And the individual, whether in a whole population, like those 1 million Americans who volunteered, or an individual who enters a clinical trial, or even an individual who presents with a disease where we want to know what the genetic basis of the disease is. Without the individual and without the clinical history, it's very hard to interpret the data. We then collect the data by sequencing the DNA or sequencing the tumour DNA, and we couple it with the clinical history, And then we get a digital footprint of the data, which is the entire genetic sequence or the proteomics. We put it together with a clinical history. Now, one of the big things is the curation of the data and the data analytics. So curation of data is, I told you there are all those genes. A lot of those genes, we don't know what they do. And a lot of those genes and the gene function, we haven't worked out. So how do we decide in that massive data what it's all doing? And that's called curation. It's kind of like this massive library where you put all the important books in one section so they're available and you can work it out, and gradually you work out what they all do, and then we can put it all together like some gigantic jigsaw puzzle. And then once we've curated the data and done the data analytics, we can predict the risk by these variations and say, well, you've got that variation, that predicts for the risk of Parkinson's disease, or that predicts for the risk of lung cancer. So what do you want to know that? I would argue you don't want to know it if there's no treatment. I don't want to know it. If there's a treatment, yes, I want to know so I can do something about it, but if there's nothing I can do about it, don't tell me. I'm not going to ask. I'm not going to get sequenced. So then there's predicted risk reporting, which is an art in itself. So, I love this cartoon. So, you may not be aware of this, but you can go online now and you can get your DNA sequenced. And actually, one of my PhD students' father is running a business where he says, I will sequence your DNA, I will tell you that you shouldn't be taking codeine or you shouldn't be taking this medication because you're more susceptible to it. So you can go online anywhere and start doing that. So it's interpret your own test results today. Patients are already doing that. Individuals are already doing that. It's whether you can interpret the data or not, but they'll give you an interpretation. But it's understanding what the pros and cons of the data are. So Monash University, we're running I'm not doing it, but some of my researchers are doing. I'm running the largest clinical trial in Australia's history. And what the really good thing about it is actually funded by the US, the NIH. It's a $75 million USA trial, which is a precision medicine trial. And it's run by John McNeil and the team in the School of Public Health at Monash University. It's called the Esprit. It's the aspirin in reducing events in the elderly. They got $50 million from the USA to run the largest ever clinical trial in Australia. And it's looking at whether people over the age of 70 benefit from taking aspirin. So as you may or may not know, there's a lot of evidence to suggest that aspirin reduces the incidence of stroke and heart attacks, but also has side effects, which is increased risk of bleeding, upset stomach, other side effects. No one has ever asked the question, should you take aspirin and you will decrease your incidence of heart attacks and stroke, or should you not take aspirin? So John has recruited, I think, up to 19,000 Australians who are 70 years of age and older, and he's put half of them on aspirin and the other half not on low-dose aspirin. John is 69 himself. He's running the largest clinical trial in Australia. And I said to him, John, are you taking aspirin? He says, no, the results aren't in, Chris. We can't do it. We can't take it. So this trial's been going for 6 years. He's not taking an aspirin yet. He's still waiting for the results to come in. I keep saying, John, what if you have a heart attack? You'll regret not taking that aspirin. He said, but I could have got an ulcer. So the moment he starts taking aspirin, I'm taking it too. So he's actually now in partnership with the Garvan Institute, is now getting the genetic sequencing of all these 20,000 Australians. And he's going to look to see, are the ones on aspirin, do they have he's really looking to see, not do I live longer, but do I live healthier? So is there a decreased incidence of Alzheimer's disease? Is there a decreased incidence of disability? Is there a decreased incidence of stroke? Now, one of the big things I think he's coming up with, he keeps hinting to me, but he won't tell me the truth, is that there is some evidence aspirin also decreases the risk of colon cancer and other cancer because it has an anti-inflammatory effect. So he's also looking to see if this cohort of Australians who are on aspirin, and he doesn't know which group are on aspirin and which aren't because it's a double-blind trial, which means it's all randomised and it's double-blind. So the outcome of this trial is aspirin shown to be overall benefit. Millions of healthy Australian older people around the world will be advised to take aspirin, and I'll be one of them. And if aspirin is shown not to be a benefit, then they'll stop taking it because it does have side effects. Now, in addition, Professor Sophie Zungas, so this is precision medicine. This is a precision medicine trial. So it's looking at a large cohort of individuals. It is checking their genetic data. It's actually called what's called an interventional study because half of them are getting some treatment and half of them not, and then they're going to look at the long-term outcomes. So Professor Sophia Zungas just got awarded last year the largest ever NHMRC grant to do a similar study, but this time look to see cholesterol-lowering agents, statins, in over 70s to see if they make a difference. So this study in aspirin is going to go for 7 years, and she's going to do a similar thing with cholesterol-lowering agents. So if you're over 70 and you have a high cholesterol, will you live longer, or will you live healthier, or will you have side effects, more or less, if you go on statins, anti-cholesterol lowering agents? And that's called the STAR-E trial. So this is our precision medicine initiative with a clinical trial, and they're going to look at the genetic background of the individuals with the biomarkers with the long-term outcomes. So I've talked to you about doing large population studies, sequencing the DNA and looking at long-term outcomes of millions of people or thousands of Australians. But what about the individual patient who comes with a diagnosis? So what does precision medicine mean to the individual if you go to your doctor tomorrow? So what I'm going to concentrate on now, because I'm a haematologist and a blood specialist, I'm going to talk about two things. First of all, I'm going to talk about pharmacogenomics briefly, and then I'm going to talk about cancer because that's my interest and my research. And it's also the best example of precision medicine. So up until a few years ago, if you presented with a lump to your doctor or a cough or a mass, they made the diagnosis by basically taking a biopsy. For some things, skin, it's easy to take a biopsy. Blood, easy to take a biopsy. Bone marrow, easy to take a biopsy. Lung, tricky. Liver, not too bad. Brain, you've got to have neurosurgery. So the way they made the diagnosis is to take a sample of the patient's tissue, look down the microscope, and just see what it looked like, basically, which is called pathology. And then, fundamentally, the same therapy is given to a whole population with that disease. It's not precise, it's reasonably accurate, and it's quite effective. And you often get either surgery to remove the whole lump or a combination chemotherapy. Now, in my specialty, which is blood, it's been very successful because blood is always accessible. You can always take blood. And because you can take blood, you can sequence it, and haematology is miles advanced of the rest of the specialties because you can sequence it and work out what's going on. The other advantage of haematology over other cancers, and this is why childhood leukemias have done so well and are virtually curable, and a lot of the adult leukemias are now have massively improved outcomes, because in general haematology and blood is often the mutations only in a single clone of cells, and so you've only got one population of cells, and therefore we can work out the molecular basis of the disease and then get early treatments. Cancer is more complicated, but there's been massive improvements in therapy because of precision medicine. So in a new way, and this is from the National Cancer Institute in the USA, we take a biopsy, and I'm going to talk to you also about a new development in precision medicine, which is actually getting tumours from the bloodstream, the tumour DNA from the bloodstream, and then instead of looking down the microscope, we sequence the tumour to look for the genetic alterations. And then we do massive mass spectrometry or proteomics or microarray to work out the genetic basis of the disease. Not what it looks like down a microscope, although we do that as well, but work out the genetic basis to design targeted therapies. So here's an example. I'm going to talk to you about an example which is chronic myeloid leukemia, which is the absolute hallmark of precision medicine. But there are all these other examples of precision medicine, including infectious diseases, metabolic diseases, but a lot of these are relatively rare diseases. I'm going to talk to you briefly about pharmacogenomics. I love this slide. The thought that serotonin uptake inhibitors are antidepressants, and the thought that you could, go off your antidepressant by looking at art seems to me vaguely appealing. And if we can come up with that after sequencing millions of individuals, I think that would be a great outcome. All right, so there is a real industry now in actually sequencing individuals' DNA, looking for gene variations that can make you more susceptible to certain types of drugs. So it's estimated in the US that 18% of 4 billion prescriptions per year, those individuals really should have an altered dose based on their genetic background, because they either metabolize the drug differently because of the genetic background, or they are modifying the drug's uptake because of the genetic background. And there's probably about 20 genes that are normal genes that don't affect you, except that when you take a particular drug, whether it be codeine or warfarin or other drugs, simvastatin, interferons, clopidogrel, which is used after an antiplatelet agent used after myocardial infarction, you metabolize them differently. I was quite shocked when I told one of my PhD students I was giving this lecture. He said to me, "Gosh, my father runs a private business in which he offers to sequence people's DNA and work out if they're sensitive to drugs or not." And I went to the website, and he does. So you can get these 20 genes sequenced and find out if you've got gene variation, and that maybe you shouldn't be taking the same dose of codeine or the same dose of clopidogrel. This is a massive industry out there. And I said, "Well, how does he know what to tell the individual?" And he said, he sends him a report saying you should decrease your dose of this particular drug. They're not going to the local doctor. So this is called pharmacogenomics. I like this slide. Here, this man is looking to a cupboard. He's looking at his medication, and this one says side effects. So I'm just going to finish by talking about targeted therapies in cancer, which is the big hope. And I want to give you the classic example of precision medicine, how we use it in haematology. So in this case, a 21-year-old woman presented to the outpatient clinic with abdominal pain. She had no findings on physical examination, and they did a blood count, and it looked pretty normal. She was mildly anemic. White cell count was normal. But her platelet count, these are the small fragments of cells that make your blood clot, was massively elevated. It was 10 times normal. So as a haematologist, I would look at that and say, she's either got iron deficiency anemia or she's got essential thrombocytosis, which is a condition where your platelet-making cells make a lot more platelets. It's a type of chronic leukemia. Look down the bone marrow. There's lots of these pre-platelet cells down the bone marrow. I'd say clinically, and based on that pathology, she's probably got essential thrombocytosis, which is a relatively benign condition. However, we haematologists don't just look down the microscope anymore. We sequence, and we do look at chromosomal arrangements. And when they did the genetic testing, she didn't actually have the JAK2 mutation, which goes with essential thrombocytosis. She had a rearrangement of the BCA-ABL chromosome, and she had chronic myeloid leukemia. So she had leukemia that would have been called a benign condition because we looked, I didn't do this, but this was from a paper, looked to see what the genetic change was. Now the advantage of blood is you can do it there. You can get the single cell and you can work it out. Now this is the most classical example of precision medicine. It is the best example because this is curative, because it happens in every cell in the white cell, this genetic change, and they designed a drug to treat this, and it's curative. But it takes a long time. So this was first discovered by Janet Rowley in the 1960s, that this chromosomal rearrangement occurred in this type of leukemia. And it took them 30 years to work out what the gene was, what the protein did, design a drug to inhibit it, and it worked. And it is this classic example of chronic myeloid leukemia has made the whole rest of the cancer field go, if we can do it for this, we can do it for anything. And it is happening, but it's complicated. So it takes a long time. It's fine to discover cures, but remember the chronic conditions are our bread and butter. So precision cancer or precision oncology. This is basically the recognition that cancer is a genetic disease. Cancer arises because you've had damage to your DNA, which makes that damage to the DNA makes your cells grow more, and they grow uncontrollably, and then they eventually spread. Now, some cancers don't spread. A lot of cancers are cured by surgical excision if you get them earlier. So the identification that is a change in the DNA means we can work out what that change does to a cell and then target therapies to correct that DNA damage or the effects of that DNA damage. And there's been remarkable success in genomics-driven cancer therapy, and it is the hope for the future, but it is very complicated, but it is working. So one of the first things you need to understand is that cancer is a genetic disease, but the number of changes that you see in your DNA will depend on what time you're looking. So this is the classic Vogelstein's hypothesis. And it's a series of colon biopsies looking at the progression to colon cancer. Colon's very accessible, so we can get lots of biopsies and see what happens. So here's the normal colon, looks pretty good. You can get a small polyp. If you looked at that small polyp, you might see one mutation in a gene known as the APC, adenomyopolyposis coli gene. If you surgically excise that polyp, you'd be fine, nothing would happen. The rest of the colon's fine. If you don't go to the doctor, or you probably don't know it's there, it grows a bit more, you get more mutations in the DNA because APC drives increased proliferation, less time for DNA repair, more mutations accumulate. Mutation now in RAS, maybe you've noticed bleeding but you don't go to the doctor. If you had that excised then, you'd be completely cured. But by the time you've got cancer of the colon, you might have mutations in 10 different genes. So if you're going to use targeted therapy, you really want to treat it at this stage, not at this stage. So the genetic errors accumulate over time. So the number of mutations you see in many cancers will depend on when you biopsy them. And so targeted therapy, when you've got multiple genes mutated, is much more difficult than if you've only got one, which is often the case in hematological malignancies. So this, you can't read this slide, but it's basically listing how many mutations, say for example in lung cancer, average number of mutations and number of genes, it'd be 163 genes in lung cancer might be affected. So it's just showing that often cancers have many different genes affected. Now the second thing to think about in cancer is that if we take a biopsy of a lung cancer, it really will depend in a way where you biopsy it. You know, if it's a small biopsy, you may miss bits with different genetic mutations. It's called tumour heterogeneity. So you might take a core biopsy here and you'll get this piece of cancer, and a core biopsy here, this piece of cancer. So it may have a mixture of cells with different mutations in them. If you take the whole sample out, then you can analyse it, but you really need to sequence all these individual different cells, ideally, to find out the range of mutations. So one of the exciting things that is happening is how do we, and this is not in clinical practice yet, but it's coming, is how do we avoid the biopsy? How do we avoid the lung biopsy or the colon biopsy? Because one of the issues that we face, say for example in breast cancer, we don't keep going back and biopsying the patients all the time. So how can we monitor the patients so they'd be like, so we can find out if they've got tumours by just taking blood samples? So one of the big things in the cancer field is we are starting to be able to detect circulating DNA from tumours in the bloodstream. And because the technology has become so advanced and we can capture that circulating DNA and we can sequence that DNA, and the studies suggest that that circulating tumour DNA that's released from the tumours matches the DNA in the tumour itself. So this is coming. In long term, I predict, and I could be wrong and I'm not going to put money on it, but I predict that in 30 years' time or 20 years' time, you know, the days of actually doing open lung biopsies or brain biopsies will be over. We'll start detecting it in the bloodstream. We'll start monitoring in the bloodstream, and we can follow the patients much more closely. So this is a very new field, and they're called liquid biopsies. And you can avoid repeat tissue biopsies. And this field is now exciting field where we're actually looking. So why does this happen? This happens because actually the cancer cells will die and they'll release the DNA, and the DNA will go into the bloodstream. And the DNA in the bloodstream matches the DNA in the tumours. So the mutations that are in the tumour will be the same, DNA will be released, and the mutations show fidelity between the two. So the issue is, how do we get the technology to do this and analyse it? It's already happening, in particular in some types of lung cancer, and it's happening in other fields as well. So this is a hypothetical situation that this paper in Nature Reviews Cancer in this year described that maybe we won't biopsy anymore. We'll take a blood sample and we'll make the original diagnosis, then we'll institute surgery or other treatment because we'll be able to tell where the cancer's coming from the nature of the mutation. We'll do treatment, and then we'll follow the patient by serial liquid biopsies where we monitor for residual disease and for response to therapy. This is what I think is coming in cancer therapy in terms of, because we can sequence the DNA and look for the changes, and we can detect the differences between the tumour DNA and normal human DNA, because the tumour DNA is different. And we can also design the therapies. So the third problem with cancer is that some cancers are so fast growing and some have so many mutations that they're very difficult to treat. And cancers are very clever, they get around it. So one of the cancers, I don't actually work on this, but one I've been really interested over the last 10 years is melanoma, because 5 of my cousins have had melanoma and 1 has died from it, because they're all my colour and they go out in the sun. Melanoma is a very interesting cancer. It's got one of the highest rates of DNA mutation of all cancers. When it's completely curable if it's excised. When it's spread, say 10 years ago, it was not a good prognosis to have. But now there's all these new therapies for melanoma. It's an incredibly exciting time to be in melanoma research. I almost wished I'd done skin instead of haematology. So the discovery that a mutation in a particular gene called BRAF1 in 40% of melanomas has led to new disease treatments with BRAF1 inhibitors. But it's also been shown, and I'll show you what actually happens, is this is actually what I work on, this really complicated. So there's these really complicated switches and signalling cascades inside the cell, and this is the BRAF1 here. And you can design inhibitors to BRAF1, but and BRAF1 switches on a switch which activates RAS, RAF1, and MEK. But if you use 2 inhibitors to both BRAF1 and MEK, you get double the effect. And this is mutated in melanoma, but if you use this inhibitor to this as well, it's very effective and you don't get resistance. And the biggest thing in cancer therapy that's really exciting is immunotherapy. Now, often I used to ask myself, why doesn't your immune system recognise your cancer as being foreign? Because it is foreign. A lot of cancers, and melanoma is an example, you develop tolerance to this foreign thing because it grows up in you. One of the interesting things about melanoma is that researchers noticed that melanoma is often associated with a lot of immune infiltrate. And now there's been this development of these new therapies called anti-PD-1 and anti-PD-L1 blockade, immune therapy for checkpoint inhibitors, which is blocking that ability of the tumour to deceive the host that it's not foreign. And so immunotherapy in melanoma is incredibly exciting, and combined with these targeted drugs for BRAF1 and MEK inhibitors, it's making a huge difference. So finally, just to the end, do we really have to sequence? I think we do, but what can we do in the short term before then? And there's been this huge development which is called clinical trials in a dish. So why should we make a diagnosis of cancer and then give you chemotherapy without testing it first, without giving it to the patient? So in the last 2 years, 2 to 3 years, there's been what's called organoid and PDX development, so-called functional testing. So the patient comes in, we take a biopsy, maybe in the future we'll take a liquid biopsy, we'll sequence them, we'll do a network analysis, we'll pick the best drug, we'll predict what the best drug is based on the genomics, and then we'll say, well, the patient should have this therapy, but maybe before we give it to the patient we should test it first so you don't get all the side effects and we can find out if it's the best therapy. This is called functional testing, and it's going to be it's a current development and progress of precision medicine, and we're actually doing some of this at Monash University. I love this slide. So it's called Clinical Trials in a Dish, and it's basically to actually test, in particular, it's a cancer screening method, is to take the tumour biopsy, to dissect it in a dish, to get out the stem cells, grow the cancer stem cells in a dish and test all the drugs on those tumours growing in a dish rather than test them on the patient. Then you don't get the side effects and you can pick which drug you like, and when you match it with genomics, then you can have functional drug testing without actually giving it to the patient. So, this is a complicated slide, I'll skip this one. Oh, here we go. So, these are called organoids, and you can grow them in a dish. They come from a tumour. You can store them and biobank them and take them out later and test them later and look at how your tumour changes. You can disease modelling, you can sequence them, and you can do host microbe, and you can do drug testing. So Professor Helen Abard and Professor Paul McMurrick are doing this at Monash University, but other sites are doing it as well. It's called precision cancer medicine, and they're doing it for colon cancer. So colon cancer is completely curable at stage 1 and stage 2, but by stage 4, when it's spread, it's difficult to treat. The current treatments are surgery, initially chemotherapy and then radiation therapy. But the chemotherapy is not really targeted. So how do you give the right drug to the right patient? So what they are doing is, Paul McMurrick's a colon cancer surgeon. He does takes out more colons than anyone else in Melbourne. I don't know if that's a good thing. He's a bloody good surgeon though. So he takes out the colon. Helen then goes and dissects out the tumour cells. And then because she's got this complicated, the technology for growing tumour cells has really improved. So we normally grow cells flat in a dish, which is called two-dimensional. But she and others have been able to grow tumour cells so they form a three-dimensional structure and they actually start to form a tumour. And you can actually grow them in a dish. And then you can say, well, I'm going to test all the chemotherapy on my tumours growing in a dish and see if they're sensitive, rather than giving it to the patient blind. The patient gets the side effects and it doesn't work. You can pre-test for it. Now, the Children's Institute in Sydney is there's a precision medicine initiative for all childhood cancers, and every childhood cancer is going to have this over the next couple of years. For all children diagnosed with cancer are going to have either tumours grown in a mouse and tested for chemotherapy in the mouse rather than the children, matched with genomics, or tumours grown in a dish. It's been funded by the federal government to actually do it for solid tumours in children. See if I can get this to work. This is colon cancer. Take the biopsy out and you grow it in a dish and then test it. So far they've done 75 normal and tumour samples. They've got 80% success rate for actually growing the tumours, and they're now starting to drug test them. So that's 75 individuals have had their samples tested, and we can test them, sequence them, and then match outcomes and also test for chemotherapy and other drug in a dish rather than in the patient. So in summary, precision medicine is very exciting. It's a very exciting time to be in medicine and also in research. It offers great hope for the future. It's not easy and it's complex, but I think it's something that we're all going to have to come to terms with and understand what it means to get the best out of it. So thank you.